Hi, I'm Min-Seung Jeon, the graduate student of Jungwon University, South Korea, and the presenter of our poster. Our research, a practical comparison of the next generation sequencing platform, depth, and assembly software using yeast genome, analyzed the optimal assembly pipeline in de novo assembly by benchmarking 4 sequencing platforms, 6 sequencing depths, and 6 assembly programs. The trigger of our research started at the point that whole genome de novo assembly is the essential process of further genomic studies and that it is hard to select the appropriate pipeline for optimal assembly due to the unique biological sequence. So we tried to reveal the synergy between the sequencing platform and the assembly algorithm, which have static property than the picker genome sequences. This is our pipeline. We sequenced repetitive yeast genome in 4 sequencing platforms and subsampled in 6 steps and assembled in 6 ways. These results show Buska completeness of 2 assembler types, non-hybrid and hybrid. First, the completeness of non-hybrid assemblers represents assemblies that, based on PacBio sequel, got higher Buska value than that of nanopore B9. By counting the number of homopolymer in each sequencing dataset, we revealed that PacBio data had about 2-fold larger homopolymer than nanopore dataset, and this difference made the gap of accuracy in draft assemblies. However, policing results also suggested that low complete draft assembly can be complemented by the secondary sequencing platforms such as Illumina and MGI. In contrast to non-hybrid results, hybrid methods represented consistent Buska value even in low depths of TGS. However, Masca, which accepted short-read first assembly strategy in common with Wagon, represented the limitation of partial alignment-based gradient extension, generating truncated Buska values. Also checked contiguity level of each assembly. The conventional indicator of contiguity such as M50 and TIG number showed that almost all nanopore-based assembly results represented more contiguous assembly than that of PacBio. However, considering that the number of reference chromosomes is 7, both assemblies revealed kinematic contiguous which are specifically vulnerable in highly variable yeast telomere. Only Canu and Spadus, which are strict to or can't connect repetitive regions, generated more non-chimeric contiguous. The last section for analysis is time complexity of each assembler. Flyer, WTBG2, and Canu exhibit a large gap of processing time. This observation implies that TGS-only assemblers which start their assembly pipeline with TGS reads can be affected by various read lengths and cause more time complexity. In contrast to Flyer and WTBG2, Canu, which has many correction stages, had more time complexity in longer sequencing reads. Masca and Wagon, which start their assembly process with short-read-based pre-assembly, showed less affection of time complexity according to the sequencing read length. This observation indicates the contiguous extension stages using TGS reads in hybrid assembly were hardly affected by the amount or length of the TGS reads. So, let me organize the conclusion. We figured out the relationship between the character of sequencing reads and assembly algorithm. This revealed the regularity of assembly process by analyzing the results of the combination of various dataset conditions and assembly programs in repetitive yeast genome. The assembly patterns also suggest that pipelines consisting of non-cooperative sequencing techniques and assembly programs lead to the whole genome reconstruction of low quality. Overall, this represents the possibility of presenting an efficient assembly strategy according to the user's purpose, even for the novel yeast whole genome assembly. Thank you.